So my area of expertise is really in technology development. I've been involved in developing methodologies for mapping or measuring epigenetic features genome-wide. I began this really at the dawn of the epigenomic era, did some of the first ChIP-seq analysis on the nascent Celexa platform at the time, and then have developed a series of technologies to measure epigenetic features since. So we use these technologies to study both the epigenetic features of normal cells and have been involved in doing this initially as part of the NIH Roadmap program, and more recently as part of the CEEHRC program, which is the Canadian Epigenetics and Environment Health Research Consortium, which are two components of the North American components of the International Human Epigenome Consortium. And so what we're trying to do within the IHEC Consortium, or the International Human Epigenome Consortium, is generate a series of reference epigenome maps to provide a foundation from which to understand how the epigenome is patterned in different cell types. As far as we know, every cell type in your body has a distinct epigenome state, so we're trying to understand what that is. And then from that foundation, then try to understand how, for example, there's variation in the population and how these can be altered in pathogenic ways in the context of cancers. My lab studies cancers that are driven by epigenetic modifications. We know that these epigenetic modifications are responsive to environmental stimulus. We know, for example, that there are epigenetic changes associated with aging, with nutrient uptake. We heard some of that today in some of the talks. But my lab studies in kind of the extreme case in cancers where we actually have genetic mutations or lesions to the enzymes that control the epigenome. So this is kind of the extreme case where we're trying to understand, even when we knock out a particular gene, what are the changes that occur? And so, for example, we study pediatric cancers that have mutations to members of the SWI/SNF complex, or the mammalian BAF complex, that's responsible for positioning nucleosomes. We study leukemias that are driven by genetic mutations to enzymes that control DNA methylation. And ultimately, what we're trying to do is understand how these epigenetic state changes can drive the initiation and progression of cancer. So I think on the technology side, I think the big, as I think we're in 2019, the big change that's coming is moving from bulk analysis to single-cell analysis. And my lab has been involved in developing single-cell methylation analysis, and we're trying to use that to essentially identify subgroups of cells based on their DNA methylation profiles. So I see that as really revolutionary in the field. I think this is across a broad range of obviously assays, RNA sequencing, ATAC-seq, and in our case, DNA methylation. So I see this as revolutionary. I think the challenge in the field is that this data is still very sparse. We're challenged by stochastic data loss, and so we've been trying to develop methodologies to group the cells based even in the sparseness to provide subgroups that we can then study in more depth once we group them in silico. And I think this is generally the approach that people are taking, but I think in the future, as we're able to be more comprehensive in our measurements, that's the route that we should be going.